I'm Ayana.
I'm Jingjing.
I'm Sangeetha.
Okay. For the first part, it's a picture of, it's a toy factory, and there's 3 workers separated by 3 walls, and they represent the cells. And so they're monitoring the amount of toys that go into the bucket. And so the rival on the far right is kind of like a silver in general. And so he poisons them first with the poison to the touch, like powder, I guess. And then it affects no one, so no one's hurt and it has no damage on them. And then in the next scene, he poisons them using gas, and it affects one of them. And so one of them dies. But the toys are still being processed, so it didn't affect them too badly. And in the last scene, he is making food poisoning, and they all end up eating the food. They all die, and it's the most effective way to kill the rival. And I should mention that in all 3 of the scenes, they're representing different scenarios in different points of time. As you can see by like the clocks in scenes 3, 5, and 8, I think. They all have clocks, and they're all showing the same time.
So basically, like the whole metaphor is that like the little cell or the workers are each individual healthy cells, so the experiment we did on biotoxicity is that we had 3 different samples of yeast cells, and we exposed them to like silver powder and silver nanoparticle and silver ions. So for the first part in this drawing, this is the little poisonous things to touch are like the silver powder. So the silver powder is too big, so the cells like recognize that it's too big and it's like dangerous, so they reject the powder from going in, so none of the workers got killed. And the second part, this whole row is representing like the whole silver ions. So like the poisonous gas, because ions are way smaller than the silver powder, so it's representative of like poisonous gas. So it's effective, but not effective enough to kill all of them, so only one of them dies, but like the production level goes down. So the way we measure how healthy the cells are during our experiment is by using like a pressure sensor. So the numbers are basically the same thing as what we did in our experiments. And the last one is what happens when we expose it to nanoparticles. So it was like extremely effective in that like it killed all of them. And when we did our experiment, like there was no change in carbon dioxide levels. So that means like all the yeast cells were basically dead. And yeah. Also, the different ways that the ions go through into the cell is by going through like the protein pores. So like the cells think that the ions are food, so that's how they take them in, and then the nanoparticles are small enough so that they go through endocytosis, so that's how the nanoparticles enters the cell.
Okay. So in this analogy, we saw a lot of things that we liked, but we also saw some limitations. So things that we liked were we liked how we saw the different levels of exposure of poison towards like the workers. It kind of showed the different levels of silver powder and the effect that they the silver and the effect that they have on cells. We also liked how they measured the toy production to kind of measure to show like how which was analogous to like the health of the cell. So yeah, and a limitation that we saw was that like the different levels of poison, they don't exactly show like the actual scale of how the difference between like the ions and like the powder and the nanoparticles. So yeah, thank you.
Okay, so audience, you have now seen these experiments once before. And so now you are more educated about, so I should point out, so they only did with 5 days, they did 5 experiments, and there's 11 groups. And so you're gonna see all of them twice and one of them 3 times. And so as we go through them more multiple times, you're more educated and you can ask even more questions. So the fact that you've seen it before does not get you out of asking questions. It makes you have less excuses for not. So the floor is open for questions.
Nicely presented and illustrated, but you said the production of a factory was representing the health of the cell. And if that's the case, I wondered how in your experiment did you measure health of a cell? Was it just whether the cell was alive or dead, or are there different degrees of health?
Okay, so the way yeast work is that they take sugar and then they like feast on it basically, and then they produce waste or like carbon dioxide. So we connected to like a tube with water in it. So the more it produces carbon dioxide, the more it pushes the water upward, and we measure like how much it pushes the water up as like if they're alive or not. So yeah. Does that answer your question?
Okay, so then go one step further. That was awesome. What is the specific analogy between what these guys are making and what the yeast made, and how are those products the same or different?
So basically, the amount of production of toys represented the amount of production of carbon dioxide. So in the silver powder, it's not able to get into the cell, so the yeast is still able to produce carbon dioxide, just like they're still able to produce toys. And the ions has some effect on it, but not as big as the nanoparticles, which is why it's still able to produce some toys or carbon dioxide in the cell. And then with the nanoparticles, it's not able to produce any toys.
Okay, but so then what is the one... what would be a limitation to that analogy in terms of how is production of toys and production of carbon dioxide different?
I guess like maybe like when they're exposed to the nanoparticles, they don't...
No, just as a product.
Maybe because carbon dioxide is waste, and toys are like...
Yeah, so one is actually a product they're trying to make. Carbon dioxide is actually a waste product. And so the issue here is whenever you make an analogy like this, and these are all beautiful analogies, they can't capture everything. So in one case, it's a waste product, and in the other case, it's a purposeful product. And the point of pointing those out is if you're teaching somebody who knows no science and nothing about the system, then people take analogies very literally, and they'll say, oh, well, so then the cell stops producing something useful like toys. Well, actually, in the experiment, this cell just stopped like breathing. So it stopped producing a waste product. And so it's worth pointing out where the analogy just isn't exactly like the experiment, because they will always be places where it is not exactly like the experiment. Okay, that was a great question. Next question. So there's some new parents here, and I thought I would just mention to all the new parents that the rule is you don't get to go to the reception unless you ask a question. Just one. So I'm trying to motivate the parents to ask some questions here. Okay, so I will ask one question while you think. Okay, so what is, so there's a lot of beautifully captured concepts here. What is a concept that is not as much captured here?
Probably like the way that the silver powder had no effect at all in the picture, but eventually like it has a little effect in the experiment because it damages the cell in a little bit of a way. So in the picture, it could have maybe shown one of the workers having like a broken arm or still being okay.
But it actually didn't do too much. There's a sort of a fundamental concept of the experiment about the differences between the things that work and didn't work that is not particularly trying to be captured here. So what is that? What is that concept? Given that this is a summer program in
Could you repeat the question?
nanoscience.
Could you repeat the question?
There's a concept that is not trying to be captured here, that is not particularly well captured in this cartoon. That's sort of fundamental to this experiment. What's different about the different things that do and don't work?
Maybe like how in each case they're damaging the cell in different ways. When in the experiment we damaged it in the same way, but more and more.
Or like, would it be like the concept of like the color and kind of?
Well, I mean, those are all good concepts. Since this is a lab on nanoscience. What concept might not?
So the scale of the powdered nanoparticles and ions.
Just the concept of size.
Yeah, size
is not what's in here, and this the thing that differentiated. And like I said, you don't capture everything. This captures a lot of beautiful things, but that's something to point out, is that the whole idea of size is not really the distinguishing feature. Whereas in the last analogy, size was the thing that it was trying to capture. Okay, so the different cartoons will capture different aspects, but just point out the ones they don't capture. Okay, one more quick question. Students, you guys get to go to the reception anyway because you worked real hard all week. All right, we will thank these guys. Who is the author of this? And is there anything you would like to add? Because there's a lot of detail here. Okay, good job, everybody.